Okay, see if you can get back in. Sorry, guys, it'll be just a second. I killed everything.
I want that recording for sure. I think your speakers are. All right. Can y'all hear me? Okay. I am going to put this. These speakers, I think, are still on. I'm trying to figure out how to shut it off. Can you hear me still? Okay. There we go. I think you can hear me now. Let's use the chat function as well if there are questions, but I definitely want to say hello to everybody in here. Thank you for having me, Dr. Panasic, and Claflin University, my alma mater. It feels so good to be walking these halls all over again, in particular to talk to you students, now as a principal scientist at Novartis Biomedical Research Institute, in San Diego, California. I really get to talk to students, so I'm very excited to speak to you guys today. But yeah, this is my guest seminar. And for today, we'll be using not only the chat feature on Zoom, but also Stimulator on all platforms. So I'm on Twitter, also known as X, Instagram, as well as stimulator.com, as Dr. Panasic mentioned. And to keep y'all entertained today, but also keep y'all actually actively listening, I'll be asking some questions throughout, and I want to keep track on who is answering the questions. So at the end of this seminar, in about 45 minutes, I am going to give somebody $50 on Cash App. Okay, and so the person who I give is the person who answers the most questions at the end of the seminar. So you got to pay attention. And the way I'll keep track of you is if you go on Instagram on Simulator, you see the handle there. I have a question that's already up right now. You just have to put your Cash App in there based on how many times you answer the question. And at the end of this hour, I will give you $50. All right. So thank you again to the host. I am a principal scientist, as I said, at Novartis. And in particular, I'm in the structure biology group. And I am currently the head of the structure-based design group in San Diego. And we focus on making medicines from scratch. And particular medicines are proteins, which I think are interesting to this class in particular, to Dr. Panasic, but also in the world. So I got my PhD as well as did my, I got my PhD in the Baker Lab at the University of Washington. This is in Seattle, Washington. And I was a postdoc at the Institute for Protein Design. This was sponsored by HHMI. This is what Dr. Panasic said in my intro. Now, what is the Baker Lab? Okay, I'm just showing some highlights that have come out of the Baker Lab in the last few decades, honestly. And this kind of gives you an idea of what my PhD is in. And before we get too much into it, I want to make sure that everyone is understanding what we're talking about here. When we talk about medicine, what is a medicine? Can somebody unmute and answer?
It is a therapeutic.
There are no wrong answers here. What does a medicine do, or what is a medicine? Feel free to also use Google, you guys. I don't mind. Again, this is all on social media, so use Google if you're doing that, but I definitely want you to interact and talk. So come on, let somebody answer. What is, let's see, I'll just call somebody at random. Desire, I see you on camera. Hi.
Hello. Yeah, I hope you can hear me.
Yes, but I can hear him probably on yours, right? Oh,
no,
oh.
Turn my
Okay, let me turn it on. Okay, can I hear you? Say hello, Desire.
Yes, can you hear me?
Yes, I can hear you, Desire. Desire Move, right? Oh, I recognize that last name. Okay, all right. Yes, so what is a medicine?
So a medicine is basically a biochemical component that is created to effect a change in a biological system. Can be organisms like humans. So
that's correct. That is correct. That is a medicine. It affects some sort of change. More, Cauldron Moore. Am I saying your name right? Yeah,
Yes, sir.
yeah, I'm
I
saying your
was
name
just,
right? Okay.
yes,
Okay.
sir.
So, yeah, what is a medicine?
A therapeutic developed to like Desire said, to onset biological changes within an organism
Yeah.
for beneficial purposes.
I like that. I like that. Okay. And then let's call somebody else. Who wants else to go? Just unmute and start talking. You don't have to turn your camera on. It's okay, but unmute so I can hear you. Or should I call somebody up? This is $50 on the line. Oh, you all go rich? Y'all are rich? Y'all don't want no $50? Oh, wow. Okay. All right. Well.
All right. Hello.
Okay. Who's talking?
Hello.
Say your name.
Hello.
What's your name?
Glory, glory,
Glory. Hi, Glory. Yes.
glory.
What is your medicine?
Medicine, in my own understanding, is something other than food that is taken that helps in healing and restoring the structural organs of the body.
I like that. I really like that definition. Yeah, other than food, and something that you take into the body to restore the normal function of the body. I like that. Okay, so there are two last people. Now, don't repeat what you've heard, okay? I want to hear what kind of substance is a medicine? What kind of substance does it have to be? Let's speak in a science term. And Glory, feel free to switch off your camera, but we'll definitely come back to you at the end of this. Now, we have two people left. Who wants to start? Or do you want to use the chat, depending on where you are? Oh, one person dropped off. Nyamongo, do you want to use the chat? Let me know what kind of substance is a medicine for it to be called a medicine. If you can't switch off your switch on your mic, it's okay. Type it in the chat. Let's hear what kind of substance does it have to be called a medicine? Okay. Well, so I'll wait for your chat at some point in this talk, but we won't wait for too long. Okay, we're gonna continue. So using the chat feature from now on, I'll be asking questions. Everybody just type in the chat. I want to see the answers, and that's how we're gonna tally this. Okay, we need to keep going. That's how we're gonna tally this. Use the chat feature on your Zoom or Instagram, and I will cash app you $50. I don't care where you are in the world. I will make sure I get you $50. I'm originally from Zimbabwe, actually, international. So, hey, Western Union, have you heard of that? So let's go. All right, so let's go. So I'm going to share these slides real quick again, and then we'll do this. Now that we have an understanding of what a medicine is, some substance that you take into the body, and it helps restore the function back to its normal state. Can you see my slides?
I can see your slides, but it's the percentage.
Oh, I'll put it in full mode. Okay. And so at the Baker Lab, they've been interested in particular on making proteins from scratch. And these are new medicines that are particularly proteins. So medicines can be different molecules of different varieties. My research focus and at the Baker Lab was specifically looking at proteins. And proteins are molecules that you all should be familiar with at this point. These are machines that mediate different biological functions, not only in humans, but in animals, in plants, in bacteria, in viruses. Proteins are fundamental through all biological systems in the universe. And so I'm just showing some highlights right here of some of the proteins that have been made de novo, that is brand new molecules, right? Brand new proteins made from scratch on the computer, tested in the lab, but now out in the world have been made from the Baker Lab. First, I'm showing right here on the, if you can see my, let me put my laser pointer on here. There's this paper about completely different protein fold, right? That was discovered and made from scratch, engineered in the Baker Lab. This is called Top 7. This was done by Brian Kumo et al. And this was work in the Baker Lab. This was around 2003, that time. Since then, they've been able to make thousands of designed proteins and structures. In fact, I'm showing here an image from a paper, my paper, actually. I was included on this paper with Gabe Rocklin, where we made literally 2,000 proteins, and all of them shaped in a specific function, 4 of which we actually characterized and showed that the shape that we designed matched exactly what the protein looks like. This was in 2017. So all the way from 2003, 2017, the Baker Lab has been making proteins of different shapes and sizes and introducing new sequences that have never been seen by nature that fold completely into the literature and database. And obviously, this all falls under the field of biochemistry and more specifically, structural biology, right? And so when we're talking about structural biology, that is understanding the form and shape of something that is a molecule, in this case, a protein, has a direct impact to its function. Now, a really great example is, let's say you want to carry some water from one room to the other. Would you use a cup? Would you use a paper cone? Would you use a cylinder? Whatever you're trying to do right there, that dictates what you're going to choose, right? So the shape or form dictates the function and vice versa, right? And so people try to understand this in different ways through taking pictures of proteins, natural proteins. This is a simplified version of talking about X-ray crystallography, if you've ever heard of that, right? Or people look at proteins in solution, like NMR. You have an NMR facility right here, nuclear magnetic resonance, right? People do that. These are all structural biologists. In the Baker Lab, we are doing it through computational prediction. So the question is, what are the amino acids that form proteins, right? And so proteins are made up of amino acids. How many amino acids are in the world? Can you type in the chat?
20.
20, that's correct. Yes, 20 natural amino acids, alanine through valine, right? Or through tryptophan and valine and tyrosine, it's 20 in total. Those are mixed in different combinations to have amino acids, right? And so can y'all name some different type of proteins on the call? Who said 20 for the amino acids?
Lysine.
Lysine is a different amino acid. That's correct. Who said 20 for the amino acids?
Caldron.
Caldron. So Caldron and Mue, you guys have each 1 point, right? All right. And so let's see who at the end of it has the most points, who will get this $50. All right. So name different types of proteins.
Are we naming actual protein structures or
Just
amino acid?
No, just a protein that you know of. For instance, I'll go first. Saliva is a protein. Saliva amylase, right? That's the enzyme that breaks down food, starchy foods in your mouth so that you have energy, right? Antibodies that fight against diseases, these are proteins. Hair on your head that's made up of keratin is a type of protein. These are all types of proteins that exist in the world. Give me different types of proteins or names of proteins that you know
of. Maybe keratin. The protein,
keratin.
the structure that makes up shellfish shells. You have.
I see what you mean.
No, chitin. Chitin. Not keratin, but
Chitin
chitin.
is not a protein.
No,
I like how you're thinking.
Hello?
I like how you're thinking. No, I like how you're thinking. That is
Okay.
great. That is in the family of molecules,
Hello,
right? And the different types of molecules. I don't think chitin is made of amino acids. You can prove me wrong, but I don't think it's made of amino acids. I think it's made of carbohydrates or what's it, sugars essentially, right? Yeah. So you are in the area, but it's not a protein quite. You're thinking of if you're thinking of shellfish, right? What shuttles
All right.
oxygen in the blood? Who's talking?
Okay, Glory, go first.
Gloria, go ahead. Yeah.
And Glory, I know we have taurine in the saliva in the mouth.
Okay. Yes, that's one. Okay, now you're on the scoreboard. And feel free, you guys, to use the chat feature on Zoom if it's not appropriate right now for you to be talking out loud. So it's okay. I don't mind. Just use the chat feature and we'll rank the points that way. Type it in the chat if you have it. I don't mind Googling and stuff like that. But yeah, we have already named a few antibodies. We named keratin. We named salivary amylase, right? And then I think is it David? Yes. David Caldron was talking about, I think he was talking maybe about what shuttles oxygen in seafood, right? And so maybe hemocyanin, that's another protein. Hemoglobin is in our blood, myoglobin, these are all different types of protein. The SARS coronavirus, now we know about its corona, right, which is a crown made of proteins. That is also a type of protein, right? So anyway, we'll keep moving. But those are all different types of proteins, and proteins mediate different functions from viruses, bacteria, humans, animals, as well as plants. Now, if you can understand the shape of a protein, right, a natural protein, that means you can understand how to make medicines, either to make sure that that shape is always formed that way, or to investigate when the shape goes bad. You can make medicines to make that shape a little bit better, or you can just make new proteins that have never existed before in nature to now become medicines. And that's what the field from the Baker Lab focuses on, right? And so we talked about different ways to be a structural biologist through crystallography and MR, but it's also computational prediction that is done in the Baker Lab. And so if you know the amino acids that form myoglobin, the amino acids that form saliva in your mouth, right, then you know that always that combination of amino acids will form that structure of a protein. Opposite, if you have a function that you want and you have a specific shape that you know attaches that function, you can find the sequence of amino acids, and that's what's listed right here. That's what I'm showing here, right here in the corner here. I'm just going to take my laser pointer. That's what this sequence of amino acids directly forms this shape right here, right? And so if I give the computer algorithm, this one in this case, this was called Rosetta. If I give it this shape and I say, I want this brand new shape that's not been existed in nature, give me the sequence
Go
that
to
will
this.
form this shape.
I
Sorry,
said,
somebody's talking. Is somebody talking? Or please be on mute if you're not answering a question, just so that we can finish throughout the recording. I think Glory, your mic is on. Did you want to say something? No? Okay. Please use the chat feature if you are in public as well. So yeah. So this shape right here, we asked the computer through this program called Rosetta, what are the amino acid strings that will form that shape? And it spits out a whole bunch of strings, and we're able to test these in the wet lab to see if they finally form the shape. And turns out with high predictability, high confidence predictability, the Baker Lab is able to do that. And so they've been able to make thousands of these proteins. And in fact, this is a specific paper I was on. And then they've done this over the years until 2020. They've done it basically using these first principles of alpha-cent unfolding and understanding natural proteins, how they fold, right? So this is using biophysics techniques. This is something Dr. Panasic teaches all the time and taught me all the time during my time here at Claflin. So I know he has taught you guys this and given you a brief overview on this, right? And so this are the principles that are used to now computationally design proteins, and they've been used over the years until 2020 when there was a breakthrough in science. It was first introduced in 2020 as hallucination, which is basically using artificial intelligence models, deep learning models, as well as machine learning models to do that same thing, predict what is the shape of a protein based on its amino acids. And then they were able to do the vice versa. Given a shape, can you use deep learning methods, machine learning methods to predict what is the amino acid sequences that will fold into that shape? And that field is called hallucination in protein design. And in fact, I will show you later on a paper that I was on where we were able to do that. And I don't know if you guys are part of the journal club here, but we'll be looking at some of these papers today with Dr. Panasic, so I hope you'll be there. Finally, I'm just highlighting here the first ever computationally designed de novo medicine that was injected into people that was made through this program. So we were just talking about what medicines are and how they are proteins in different forms. Here's an actual protein that we know is running in people right now that was designed on the computer. So this field has really grown over the years from 2003 when we were making just, proteins with no real function, but we're making just, imaginary shapes that have never existed before. And we're actually confirming that to now we are making life-saving medications that are injected in people from scratch. And this is now being largely aided by artificial intelligence models, like the ones developed in the Baker Lab, specifically at the Institute for Protein Design. It's now called Rosetta fold diffusion, also known as RF diffusion, which is a method that anyone can learn because it's all available online, which is why I really want to encourage most people to really continually use online materials to enhance your skills. This is going to be the theme of this entire talk, okay? All right, so those are the highlights, right? And maybe we can talk a little bit more about this medicine from the IPD, right? This is the Institute for Protein Design, work led by David Baker, Neil King, and David Wiesler. David Wiesler's lab focused particularly on coronaviruses and understanding the structure of coronaviruses He's a structural biologist who uses microscopy specifically to look at coronaviruses in particular. And so you can imagine in 2020, when the pandemic hit, he was instrumental in understanding why the COVID-19 was so difficult to understand and difficult to basically, kill, right? So he was instrumental in helping us design proteins that would form and help to neutralize this virus. And one protein in particular, which was made, this is the first ever approved computationally de novo designed medicine that's now injected in people, real people, y'all. This is a milestone. So in June 29, South Korea actually approved this Sky Covion. This is from the Institute for Protein Design at the University of Washington. And it successfully passed phase 3 trials with 4,000 and something adults, and generated 3 times more neutralizing antibodies than the known vaccine at that time. Antibody conversion rate 90%, 98%. Among subjects greater than 65 age. 95% of those antibodies were activated. And so, yeah, no serious adverse reactions. Heterologous booster trials are now underway to see if you can boost and generate more neutralizing antibodies. So this field of computational design, computational protein design, is currently not only making an impact in science, but it's actually making an impact in people's lives. Like, this is a protein that came from somebody's mind onto the computational screen, was designed on the computer, was tested in the wet lab, and now is currently injected in the bodies of human beings. So the IPD at the Institute of Protein Design has also made a commercial impact, right? Not just with people and not just in science, but also commercially. So I'm just showing right here, this is a screenshot from 2020. So it's not even updated really up to today, 2023, of the number of startup labs, startup companies that have spun out from the University of Washington, right? So each of these companies that have generated these amounts of money based on these amounts of these times, right? 2020, you can see PVP, which focuses on celiac disease, generated like $365 million. A Alpha Bio, 2.8 million. This is just raising funds. Remember, like, this is just, they haven't even started to actually make products. They're just raising funds, and this is the amount of money they're raising. And all of these people are structural biologists like yourself, right? In this class, these are people who are interested in making a world impact. And they are all focused on medicines. There are people who could focus not only on medicines, but they could focus on plants and agriculture, because again, proteins exist everywhere, and they can focus on things that will affect climate change, like the people in this class who are interested. But these are the skill sets that are used widely, and I'm just demonstrating that you can use them in multiple different cases, not only to make an impact in science and people, but also a commercial significant impact. Lastly, the protein design group that initially started, this is a picture from 2004. You can see this was a pretty small group right here. This was the Baker Lab going to the Rosetta Con, but it exploded over the years. In 2017, you actually see it was quite a lot of people. Again, not well updated. In 2023, it's a much large group, and you can actually see in figure 5, you can find this with this. Yes, that is me. Dr. Panasik has already spotted me. That is me right there. In 2017, I was in this group, and if you want to take a closer look at this picture, there's a paper that was actually published in PLOS Computational Biology on May 2020. You can go check that out in the citations. But yeah, this group is pretty large. This is actually David right here. That's the man himself, David Baker. And in fact, we had them over recently at Novartis for a talk. And so we're really trying to make an impact and democratize protein design across the globe. And you students in climate engineering class are going to be the future of this field. You have to get and acquire these skills. This is why this class is so fundamental. So, again, thank you again to Dr. Panasik for inviting me, but also for instilling these skills in you guys at this early stage in your career. So yeah, I was in the Baker Lab, as you saw. That's me in the purple, and this is some of the impact I was able to make into the world, right? So these are some of the papers that Dr. Panasik was mentioning early on, and so we talked about computationally designed proteins that accurately fold into the way they were designed. And this paper, actually, we were able to do like 2,000 proteins, test them in yeast surface display, and then confirm by purifying individual proteins and actually taking what, essentially screenshots, right? X-ray crystallography, NMR, to take a picture of the actual molecule and then compare it to the design. And we found high confidence and high fidelity and agreement between the structure as well as the design. In over, I think it was like 35 in total of the 2,000 that we designed and that were confirmed in the wet lab, about 35 of them were confirmed individually as opposed to just in a pool. And then 4, actually, we got structures off through NMR, and they matched exactly the design. This paper was published in 2017 in the journal known as Science. Please do check it out. I love that paper. It's one of my favorite. It was my second paper, and it now up to date is my most cited paper, and so it is quite fundamental in order to understand some of the principles of folding and design. And we use those principles to generate new artificial intelligence models that now use deep learning as well as different machine learning techniques like diffusion and several other ones to generate now models that can make proteins from scratch just using these AI first principles. And so this paper was the first indication of hallucination, which is basically using artificial intelligence to make brand new proteins. This came out in 2021 in the journal Nature. And it was pretty a landmark paper. And we were able to use both the old physics model systems as well as the new AI-based systems for protein design to make impactful proteins that Dr. Panassi was talking about earlier on, that are basically the grounds of making antibodies and vaccines and nanobodies and SCFEs in the future. And these proteins are called immunoglobulin-like domains. This is my main focus throughout my life. I've been fascinated by this particular type of protein shape and what are the fundamental rules that make this protein shape. And I imagine we'll be talking about this later on in the journal club. I hope some of you will be in there, but these are some of the papers that we'll be discussing. Please do take a look at these. If you have questions offline, even after this, please contact me at any point in time, and let's formulate some questions and some discussions. This is the last thing I'm just going to cover within this class. It's pretty short, right? You can use all of these, again, to design proteins, right, for the specific shape, and the proteins that you are interested in are based on your specific interests, right? Which is why it's important to have designers, protein designers from all different kinds of backgrounds, all different kinds of mediums, because the impact you make on the chosen protein that you have impacts not only that specific protein field, but also impacts your community, right? And so if you identify an area or a field that currently doesn't have a protein that is addressing that problem, then you get to design a brand new protein that will address that specific problem, which is why it's important to have protein designers from diverse backgrounds and diverse areas. It's one of my main missions to democratize AI across the globe. One example right here we're showing is how they, this paper right here, this is from the Baker Lab. Oh, I haven't, this is brand new, actually. I think in the last month, they put this as a preprint, actually, so it hasn't even been fully published. They published this method using AI principles that we were just talking about that to improve an enzyme as well as the binder. So one protein we were talking about earlier on that shuttles oxygen in our blood. Actually, this is a perfect time for a question. Yeah, a protein that shuttles oxygen in your blood is called a hemoglobin protein, right? But the proteins that store oxygens in your muscle, what is that protein called?
Myoglobin.
Myoglobin, that's correct. Okay, that's an additional point to you. Okay, make sure to keep me honest on Instagram or in the chat so that we know who is getting the $50 at the end. Myoglobin, exactly. It's a protein that stores oxygen in your muscle cells, right? And so myoglobin, actually, they took that protein, myoglobin, and improved its binding to oxygen. Okay, in this paper right here, they improved to make brand new myoglobins, right? Myoglobin, by the way, was the first ever protein that was characterized in structure, was characterized in the PDB. So if you go to the protein data bank, the very first protein ever that was ever characterized and got like a picture of or got a structure of, I think this was through X-ray crystallography, was myoglobin. And so it's pretty full circle moment to now be able to make many different types of myoglobin with very relatively low to high sequence fidelity to the native myoglobin. So these are now just brand new myoglobins, about 2,000 of them that they made here, that are all active in different active rates and capacities, and how well they bind to oxygen, how well they bind to iron in particular that mediates the oxygen binding. And so this is really, pretty amazing. These are things that you could do with AI-based design. Lastly, they also showed an enzyme in here that actually, what does an enzyme do? Does anyone want to say it?
So it speeds up
Catalyzes
the
reaction.
Okay, let's start with Keldra. Keldrin, let's start with you. Go ahead.
Yes, sir. Catalyzes reactions.
Catalyzes reactions. Yes, that is correct. Do you have a different answer, Nwe? What does an enzyme do? It has to be different than Caltrans for you to compete on this. Yeah.
I wanted to say enzymes, they speed up reactions without themselves being consumed.
That is an additional point. Yes, that's additional good marker, right? So both of you get a point there. So make sure to keep me honest in the chat or on Instagram stories in the answer feature. You'll see there's a question there to put your cash ups there so that I know who had the most answers. But yeah, that is a different element. An enzyme speeds up reactions, right? So that's why it's called a catalyst, as what Caltrin said. But the fundamental point of both of that's the function of the protein, right? A lot of molecules speed up reactions, but in order for it to be called a catalyst and specifically an enzyme, it doesn't get modified itself while making that change or making that speeding up that reaction. And so that is definitely a good nuance. One enzyme in particular right here is TEV. TEV protease basically cleaves a specific variant, right? And so it's a specific substrate that it actually cleaves. Does anyone know what's the substrate that TEV cleaves? Well, that will be homework for later. I'll leave you with that so that you can look up this paper and protein. And when we connect at a later date, we will talk more about this. I want you to, contact me on all different platforms, Stimulator, but also LinkedIn, and let's connect. And that's homework for the future. If you come up to me and say, hey, I know you from Claflin, we were talking, blah, blah, the first question I'm going to ask you is, what is TEV? And what does it do? If you cannot answer that, we cannot have that conversation. But they were able to improve that enzyme, particularly TEV, and show remarkable changes with that. So it's multiple enzymes now we can make on an industrial level. You can imagine what an enzyme, which is a molecule that speeds up reactions, right? If you can make brand new ones on an industrial scale, what is the impact on climate change that you can have? We can now abandon fossil fuels, right? In a way, and use biological molecules that are naturally degradable, right? Which means they don't last a lifetime, right? Use those instead of any of the specific ways that are used for catalyzing reactions currently, which are harmful to the environment by producing either gases, carbon dioxide emissions, all this kind of stuff, contributing to global warming. We can replace that now with natural amino acids, natural proteins that are made up of amino acids and break down later on. And so this is one way protein design and AI mediated protein design can directly impact climate change in a broader context. And so you already have that. But yeah, I want to say thank you so much. This is my acknowledgement slide. I want to really give a big shout out to Dr. Panasic. Thank you so much for inviting me to give this talk here. I hope I'll talk to you guys again in the future. Also, a big shout out to Dr. Harris as well as Dr. T. Harris for organizing this visit. Big thank you to them for helping Dr. Panasic through this and organizing this entire visit here at Claflin. These are some of the other technologists people I've worked with over the past, the Institute for Protein Design, as well as people that I currently work with at Novartis. And so, yeah, please contact me for any questions. I'm at Stimulator on all platforms, Instagram, Twitter, TikTok. I'm everywhere, okay? And also at stimulator.com. You will find me there doing Steam Olympics, which we're doing today. In fact, 1:30, we'll have a live demonstration of Steam Olympics. These are people who are competing for $1,500 a prize, and they started a few months ago. And we'll actually be doing this throughout this week and next week, where two students who went through Steam Olympics season 1 actually and competed, designed their own proteins, brand new proteins that did not exist before. One designed them as a haiku, so using poetry as a haiku, designed it to basically help fashion to be sustainable. The other person designed this protein using a wrap. scheme, a rhyme scheme, Get Into It by Doja Cat. She used that rhyme scheme and made a protein that will help chelate heavy metals in water to basically improve water conditions in Florida. They'll both be presenting at ABRCAMS. I don't know if you've ever heard of ABRCAMS, but annual biomedical research conference for minoritized students. It's an international conference with about thousands of students. 5,000 was last year, actually. And they will both be presenting posters there. So please do find our booth, Stimulator LLC, which will be there this year. If you're going to ABRCAMS, we'd love to see you. They also have a virtual section. So please, if you're not able to make it in person, you can also register online and come through to our booth. We will be showcasing STEAM Olympics and how you too can design your own proteins as a citizen scientist or as a protein designer in the making to address whatever specific novel functions that you are looking to do. Okay, so I will, that's everything. Did I make it on time? Yes. Yeah. Okay, that's
and just have lunch afterwards.
Yeah.
If you want to stick around and answer a couple questions, I'm
Please,
sure that they would have a ton.
Yeah. In fact, I love questions. I would love to answer some questions. So let's see. Let's read the chat. Desire, you answer that. I mean, ask the question 20. Oh yeah, so because we got to make sure who got the most answers, right? So we do that. The 50. Is it gonna be Cash App, Western Union, whatever it's gonna be? It's gonna be $50 today from Stimulator LLC. Okay, so Desire has maybe what? How many answers did you get correct, Desire?
I got all my answers correct.
Yeah. How many answers do you think you got today?
I think it was 3.
3 answers. Okay, so 3 answers, and then Caldrin, you were also competing there. How many answers do you think you got correct today?
I think it was only 2.
2. Okay, so 2 and 3 over there, and the time is not up yet. And then I think Glory was okay. She's now dropped off the call. My network is unstable, so it's breaking. Okay, so I think she had 1 question that she got correct, right? And so we got 3, 2, 1, and then if you had an answer, put it in the chat as well, so Dr. Chidyausiku can easily keep track. Thank you so much, Dr. P. I appreciate that. Yeah. All right, and then so everybody else, I guess, on here, Shepherd Gwiza, Antonio, and Nyamongo, y'all all have 0 right now, and so feel free to answer in the chat so that you enter the competition. You all have 0 in the running. The person with the most points at the end gets $50. I don't care where you are, you're going to get it. And so let's use these last 15 minutes constructively. Let's ask any questions you have. Each person who asks a question gets a point. Let's go.
So I did have a question on your research papers. I want to ask what was the overall goal of the papers? I would say in layman's terms, because after I read the abstract of both of them, I kind of had an idea, but once I got into the actual body of work, my interpretation was kind of lost because I didn't really understand everything I was looking at.
No,
so what was the overall?
Question, it's a good question. A goal that connects all those 3 papers, because each of them have different goals, of course, right? Depending on what the paper that you're looking at. But the overall goal really of a lot of these protein design papers or design projects is really to understand the fundamental principles of folding, protein folding. How do proteins attain the shape that they attain through natural processes, right? How do they do that? How can we recreate that in design, right? And that's why we do design, so that we design and understand the process while designing, while engineering, right? Because if you can make something from scratch, then you definitely understand how it's made, right? And you understand how it's attained. And so that's one principle. Another principle, which all structural biologists have been fascinated, including Dr. Panasic and the work that I did here through my 4 years here at Claflin University, is understanding, okay, the structure, how it's folded is one question, right? But how is that linked to its function, right? And so what is that link like? How flexible is that link, right? So we see my hand right here. It is 5 fingers, right? And if I grab this water bottle, it can curve and become a hand that grabs the water bottle. The function here was to grab the water bottle. That's the function, right? And the shape of my hand is adapted specifically to do that. If my hand was shaped like a cell phone, right, it can't grab the water bottle, right? It cannot grab the water bottle, right? So function and shape are directly linked. Now in proteins, the question is, how is that link? How amenable is that link, right? And you answered that question in multiple different ways. People have answered that particularly on different proteins that they're focusing on. We talked about TERF protein and myoglobin protein. We talked about immunoglobulin proteins. But here in the Panasic lab, you've talked about luciferase proteins, beta-galactosidase proteins, biodiesel types of proteins, proteins, right? And so it's a plethora of proteins that you can specifically focus your study on. And you, in the climate change case, right, I'm sure you're identifying proteins that you guys want to focus on. And it's all in the pursuit of understanding these questions. What is the holy grail of biochemistry, right? It's understand protein function and structure. But that's a really good question, Cauldron. 1 point to you. So you guys, I guess, even now, 3-3 with Desire. Does anybody else want to answer the race? You know, let's open the race for anybody else in the chat. Ask a good question and you'll get a point. Ask multiple questions, you get as many points as the number of questions that you have. We'll going once, going twice. Anybody in the chat? Anybody who's not Desire or Cauldron? Anybody? Oh, what a shame. Oh, Shepherd, you seem to unmute. Do you want to say something, Shepherd?
Yeah, I came late. It's raining in Nakuru, so I just got into the middle of the discussion. I'm not sure how everything is going.
It's okay. It's okay. That's okay. Can you ask a question of what you have heard so far? Ask a question that you are curious about in protein design. Yeah.
Or anything.
Do you understand the question, Shepherd?
No,
Shepherd,
I didn't get it. I'm still trying to grasp. Yeah, I think
Do you have any questions? Any questions at all about protein design and how it affects climate change? Any question at all? Also, this applies to Antonio. This applies to Nyamongo. Any questions you guys come up with in the chat, put it in the chat. There's $50 US dollars on the prize here that will come through Western Union or Cash App. Type in the chat. Right now, Desire and Cauldron, you guys are ahead. So let's see. Let's go neck and neck. You guys have any other questions? Desire, Cauldron, anyone?
I was looking at like some of the diseases that I have encountered in patients in the hospital, like the ones that are caused by protein degeneration, such as where in cells they demyelinate, and then, like for example, in the case of cystic fibrosis, we have proteins that are like defective proteins that are manufactured. So how would you like help someone who's trying to conduct something to like a solution to that, where we can have something either to like stop the denaturation or have like a renaturation process of denatured proteins?
That's a good question. How would that? Yeah, that's a good question. So essentially, you know, we've been talking about how proteins fold. You're asking if proteins unfold, how do we get them to refold, right? And so it's a very similar question, right? Because, yes, you know, proteins are molecules, and molecules always stay in a state of equilibrium, right? And so they're not always folded, right? They're always going back and forth between folded and unfolded states. And so your question really goes at the heart of, can we understand how the refolding process happens in the same way we understand how folding process happens, right? And that's a great question. It's one that's also addressed in protein design. The answer is, I don't know the answer. But there are several people who are looking for that answer right now. These are also called structural biologists, and they're approaching it in multiple different ways depending on the protein. But it's a good question. Another point to you. So you're in lead now. Anyone else has a question? There's a question in the chat. How did you break into the industry of biotech with it being so lucrative? Oh, yes, that's a good question. Well, I would owe it directly to Claflin University, and in particular, Dr. Panasic. I have a really fond memory. Actually, it was an assembly in what is the gymnasium, JTK, and this was after matriculation day, and I'd heard a senior Panasic Lab, a student at that time, Johnny Walker. He had been given the presidential award, award or something at matriculation day, and I was just in awe. I was like, oh my God, that's amazing. And right after, Dr. Panasic approached me, and this is the time when I think I was a volunteer in the lab at the time, washing dishes, just doing what I can to learn. And, Johnny was my mentor at that time, together with Shweta, Kariya-san. These are all Panasic Lab alumni. And at the time, I was just in awe. I was like, oh, wow. That's amazing. Johnny is getting all these prizes, for doing the stuff that we're doing in lab. I want to grow up to be like that one day, And then Dr. Panasik approached me and he literally was like, you have what it takes to be, on down that stage of winning awards one day. You have what it takes to become a scientist, to, enhance your career. And that was the very first indication that I was like, oh, wow, somebody actually believes in me in order to push forward my dreams. And that first interaction was amazing, but I've also then looked at several mentors throughout my journey, Dr. Panasik, Dr. Harris, Dr. Freddie Tisdale here, in fact, your dean, as well as Dr. McCutcheon, so many people here at Claflin University. And then when I went on to Novartis, there was Anastasia, Dr. Ward, Dr. Michael Acker, so many mentors that have impacted my life. Dr. David Baker, we've been talking about here today, where I got my PhD, Dr. Marcos, the people who I co-authored these papers with. It wasn't alone, this journey has not been alone. That's why my acknowledgement slide was so long, and indeed, even the mentors that I have now at Novartis, I have several people that I rely on, not only my associates within my team, but also some of my managers, my mentors who are currently working with. So yeah, that's really how you break into the industry. You identify a mentor who has either had experience in that industry or who has the skill set that you want to acquire or you think is in the field that you want to acquire. Identify them, impress them so that they become your mentor. Just simply ask them to become your mentor and hold on for dear life. Don't let go over years, even if after you graduate, still come and bother them and ask questions like I do with Dr. Panasik, and they will lead you throughout your career to become lucrative. Yeah. All right. And so another question in the chat. Antonio, oh, hello. Welcome to the race. Antonio has answered. Oh, so Caldron, you now have 4 points and Desire 4 points. Okay. So what protein projection can contribute to the transition to renewable energy sources? That is such a climate change question, Antonio. Wow. Okay. And please tell me if I'm saying that name correctly, Antonio.
That's his last name.
It's a Q. Okay, it's a Q. It's a Q. Okay, it's a Q. That's a really pertinent question to climate change. Mozambique. Oh, okay, right next door. Okay. All right. So, what protein projection can contribute to the transition to renewable energy sources? So away from fossil fuels, moving to renewable energy sources, what protein can help us do that? I think some of the lab that is happening, some of the research that's happening right now in the protein design lab here at, in the biochemistry lab here at Claflin University and Panasik's lab, focuses on enzymes, right? And this particular class of enzymes, which are alpha beta proteins, right? And so these alpha beta barrel proteins make up 10% of all known enzymes in nature. And so in a particular class of those alpha beta barrel proteins are these proteins related to biofuels, right? What is it called? Glycosides? Yeah.
Glycosides. There's a ton of different ones. Yeah. If you want to break down cellulose or non-food related plants into sugars that you can then ferment into a biofuel.
Love that. Yeah. And so if you guys didn't hear that because he's on mute, yeah, if you are using enzymes to break down non-food related substances like cellulose from plants or grass or something else, break that down into renewable energy fuels, right, that are not reliant on fossil fuels. Like you could break that down into ethanol or different types of fuels that you can use. That is a way to get around relying on gasoline, which relies on fossil fuels, which is not a renewable energy. So glycosides is definitely one answer to your question, Antonio. So yeah. So you got 1 point already. Okay. Oh, now we got new messages coming in the chat. Oh, people want to get into the race. They want this $50. Okay. So we'll say Antonio got 1 point, and then Desire, you got you are in there. Okay, you are global now. I wanted to know what are your plans for Zimbabwe Biotech. Okay, well, let's bookmark that. Let's go to the next one. Okay, who would you ever use your expertise to make biochemical weaponry? The answer to that question is no. And so we don't have to focus too much on that, but the answer is no. I would never do that. Shepard Gwizza says, how does beef production affect greenhouse gas emissions? Oh, that is such a good question. Yeah. You know what I will do, Shepard? Since this is a late breaking question and we're right at 2 minutes, I will say, Shepard, I will ask you that question. Let me know through an essay or a paragraph on any of my social media, right? Any of us, so contact me on the Stimulator, right? It's everywhere Stimulator. Make sure you find it. Contact me on there because I want to connect with you guys even after this. Even LinkedIn, I'm on there, or just go to stimulator.com. And if you go to the contact portion, you'll be able to directly message me right there if you don't have social media. But I am Stimulator on Instagram, Twitter, X, all that. So contact me, just write me right there. That's perfect. Perfect. Email me there, and I want to hear your response to this question. How does beef production affect greenhouse gas emissions? I have a good idea based on protein design, but I want to hear your response, and we can connect there. But yeah, I think at this point with Josiah's 5th question, you're the winner. You're global now. I wanted to know, what are your plans for Zimbabwe in biotech? Actually, is that correct? 5th question, and Cauldron just asked the question about the bioweaponry. That's also a question. Let's see. So, all right, but my plan is to get students like you to become also protein designers and be the future stimulators in the world, to move this field forward and find the novel tools and skills and use them to solve the problems that we have, not only in Zimbabwe, but in all different types of third world countries globally. And that's why it's important that you're in this class, international class of climate change. I think you guys can learn a lot of things. So I think Josiah and Cauldron, you guys are even. You each definitely contact me after this. You each win this prize, okay? And so thank you so much for joining me today. I hope you guys enjoyed yourself, and I really hope we connect further in the future.
Thanks a lot. Can everybody? Everybody thank Tamuka for a stellar talk. Love it. Love it.
Yes, sir.
Thanks a lot.
Thank you.
Okay.
Thank you
Have a
so
great
much.
day, guys. I'll send you links later on. And if you got any questions or anything like that, feel free to contact me as well. Talk to you soon. See you Monday.